Emotions and defining them have puzzled scientists for centuries. In recent years, despite many hours of research, scientists continue to disagree on its definition. One definition for our purposes states, emotions are neurological processes that have evolved to guide behavior in such a manner to increase survival and reproduction. For example, they improve our ability to learn from the environment and the past. Even this definition very vaguely describes the effects of emotion and tells us very little about what emotions really are. Another definition is that emotions are a psychological state that embodies a feeling that not only is recognizable, but also characterized through facial expressions and behavioral patterns of arousal. Despite its contradiction and vague definition, emotion has very many neurological processes to study.
Many people believe that feelings and emotions use the same neural pathways. But many researchers claim that a feeling is a subjective experience of the emotion, but not the emotion itself. These two events are dissociable, and they use separate neural systems. Recent research has indicated there's not just one neural circuit of emotion. Rather, depending on the emotional task or situation, we can expect different neural systems to be involved. One of the major areas of the brain that is involved in emotional processing is the limbic system. The limbic system is a complex set of structures that lie on both sides of the thalamus, just under the cerebrum. It includes the hypothalamus, the hippocampus, and the amygdala, along with other several parts of the brain. It plays a major role in the regulation of emotional responses, along with several other important functions. The hypothalamus plays a role in the activation of the sympathetic nervous system, which is a part of any emotional reaction. The thalamus serves as a sensory relay center. Its neurons project signals to both the amygdala and the higher cortical regions for further processing. The amygdala plays a role in processing emotional information and sending that information onto cortical structures. The hippocampus integrates emotional experience with cognition. The insula, which is associated with the emotion of disgust, and the anterior cingulate cortex are jointly active in participants experiencing emotional feelings including maternal and romantic love, anger, fear, sadness, happiness, disgust, and trust. It appears then that the insula is active with all feelings, both physical, body states, and emotional, suggesting that it may be the junction where cognitive and emotional information are integrated. The role of the insula as body information central is also indicated by its connections to networks across the cortex and to the amygdala, which is its role in evaluating emotional stimuli. The orbitofrontal cortex is activated when identifying angry facial expressions and hearing angry prosody, and the anterior insula is linked to identification and experience of disgust. The insula appears to play a broad role in integrating affective and cognitive processes. Emotional research has shifted from identifying areas that specialize in a specific emotion and characterizing how these areas interact and determining if there are any interactions common to different types of emotional experience.
We know more about contributions to emotion from the amygdala than from any other brain area. There are two amygdalae, one present in each hemisphere. Each amygdala is a small, almond-shaped structure in the medial temporal lobe adjacent to the interior portion of the hippocampus. The amygdala is best known for its role in perception of emotions such as anger, fear, and sadness, as well as the controlling of aggression. The amygdala also helps to store memories of events and emotions so that an individual may be able to recognize similar events in the future. For example, if you have ever suffered a dog bite, then the amygdala may help in processing that event and therefore increase your fear or alertness around dogs. The size of the amygdala is positively correlated with increased aggression and physical behavior.
One thing that increases our chance of survival is our brain's ability to trigger a state of fear in response to dangerous and threatening stimuli in the external environment. Over time, our brain learns and associates certain stimuli with fearful responses that allows us to better avoid them in the future. This is known as fear conditioning. Damage to the amygdala impairs conditioned fear responses. This is because people with amygdala damage lose the ability to implicitly learn to respond to stimuli that represent aversive events. And this is due to the lack of connections between amygdala and other sensory and association cortices that are required to represent external fearful stimuli. Similarly, patient SM displayed a profound and pervasive impairment in the induction and experience of fear across a wide range of situations, such as SM repeatedly told the experimenters that she hates snakes and spiders and tries to avoid them. However, when she was exposed to snakes and spiders in a pet store, her actual real-life behavior exhibited no fearful responses. Rather, she was attracted to them.
Damage to the amygdala results in an individual losing the ability to process fearful stimuli. One such case study involves patient SM who, despite being involved in many frightening situations, couldn't remember actually having felt scared since she was 10. SM was able to process the concept of fear and also able to describe situations that would elicit fear. However, because of her inability to experience fear, she often ended up getting involved in dangerous situations. Standard neuropsychological tests on SM reveal normal intelligence, perception, and motor ability, but something unusual was discovered during a test for emotion processing, where she was asked to judge the emotion being expressed on the faces in a series of photographs. So I'm going to show you some pictures of a woman, and I want you to tell me what emotion is she expressing. Okay, this is the first one.
She looks surprised. Sad. She looks kind of sad now. Happiness. She looks happy. Yeah. Angry. This one, she looks kind of disgusted. Something.
What about this one?
I can tell that there's an emotion being expressed, but I don't really know what it is. I can't tell.
The results of this experiment show us that SM was able to accurately identify expressions conveying sadness, anger, disgust, happiness, and surprise, but failed to recognize a face expressing fear. An MRI scan of her brain revealed highly specific lesions encompassing the regions of her amygdala. It was discovered that she suffered from a rare autosomal recessive genetic disorder that led to the bilateral atrophy of both of her amygdala. Thus, this experiment shows us a positive correlation between amygdala damage and the inability to experience fear.
Despite the success of relating structures to various emotions, emotion research has shifted from the study of isolated neural structures to the investigation of neural circuits and whether there are specific circuits for specific emotions. To better understand how the brain produces normal and adaptive emotional responses, we need to understand how these structures interact with each other and with other brain regions, such as the role played by the amygdala and other emotion processing structures in the neuropsychiatric disorders is not well understood. And knowing the exact neural circuits involved in processing negative emotion, such as fear, sadness, and anger, will allow us to develop better and more effective ways to treat these disorders.